I wasn't expecting to talk about lupus, if I'm being totally honest.
Yeah,
But
well, there you go.
yeah,
There you go. Yep, I'm a hammer right now.
yeah. This is Audiommunity, a podcast about our body's never-ending fight with the outside world.
Hi, everyone, and welcome back to Audiommunity. This is episode 47. My name is Matt Woodruff, and I am joined by Dr. Kevin Bonham.
Hello, hello. Don't know why you had to give me my title there.
Yeah, it's important sometimes that we all remember. In a time where nothing means anything. Yeah, you worked hard for that title, and I'll give it to you sometimes when I feel like it.
Thanks.
So, we are going to talk today about RNA. And this is actually a thing that I've been thinking a lot about over the last couple of years. And, you know, the paper that we're going to be talking about today is called RNA and Glycosylation Enables Immune Evasion and Homeostatic Efferocytosis. First author is Vincent Graziano. Let's hope that we did okay there. And Vijay Rathanam.
So this is actually a first and last author, I think.
That's a good point with Jennifer Porat.
Yeah.
So, you know, we'll talk a lot, I think, about molecular mechanisms and signaling today. I know that this is something, Kevin, that you have thought a lot about, both in previous lives and in your current one. But I actually want to start today taking a bit of a twist into this, which is the sort of TLR7 autoimmune axis, which I think is really interesting. And I'm sort of, you know, jumping the gun a little bit on the paper, because at the end of the day, they conclude with what I think is actually some of the most interesting stuff about, you know, why this might be important. This is a Nature paper, by the way, so, you know, it has received some attention. So congratulations.
Yeah, actually, in my notes, I'm going to hold it up to the camera here. The first thing that I have on my notes is, why is this a Nature paper?
Sure.
Which I thought is an interesting way to start, because, like, this is solid work, and we'll get into the details of the paper. I think going through, you sort of march through the paper. It's very good science. It's well done. But when I first looked through it, my first reaction is like, okay, this is cool, but why is this a Nature paper? Why did it go so high? And I have some theories, but I'm curious where you landed, because it sounds like maybe we have different thoughts on why this is important. But I'd love to hear what you think.
Yeah, well, let's sort of start at the basics of autoimmunity and SLE. Right, so systemic lupus erythematosus, right? Lupus, as most people just
you didn't even stumble over that last word, which I always do.
Yeah. So
erythematosus.
Yeah, you got it. So lupus is an interesting disease, right? Because it manifests itself in a lot of different ways, right? And this is basically, at the end of the day, an autoimmune attack, a systemic autoimmune attack on your whole body, right? So lots of different tissues, lots of different organ involvements, and things like this. And so there are some real challenges in understanding lupus in a human setting. And the reason, and this is true of mostly of all autoimmune diseases, part of the reason that it can be hard, and I think we've talked about this plenty on this podcast, part of the reason that it can be hard to study autoimmune diseases, particularly in people, is because autoimmune diseases progress very slowly. Your immune system is not designed to just full tilt attack, right? We have compensation. There's always a dieback phase in an immune response, and that's true of autoimmune disease as well, right? So if you develop antibodies against a thing, right, against some self-protein, you don't just continue to expand that response until you're dead. What happens is you start to develop, for example, an antibody or a T-cell response against a particular molecule, particular protein, and then that immune system ramps up and it fades, like it would in any other situation, right? And we see this in infectious disease as well. TB is a great example, right? So if somebody gets tuberculosis, it infects your lungs, right? It gets inside the macrophages in your lungs. Your immune system knows that it's there, knows that it's inside those macrophages, but it ultimately can't clear it, right? So your immune system has a couple choices. One choice is to continue attacking you, continue attacking your lungs until your lungs are just a giant ball of inflammation, and then you die. The other option is to say, maybe we should back off a little bit, right? And so what you do instead is, in tuberculosis, it's its own thing, right? You sort of wall off the infection, you die. But the important thing to note is that there is this sort of moment of dieback, right? This is why we refer to autoimmune diseases as relapsing and remitting, right? So the relapse, right, is the new effort of your immune system to attack your own tissues, your own organs, and then after a while, right, after a while of that relapse, then it remits, it pulls back, right? And so what you get are these sort of flare cycles in lupus, where somebody will be stable for some amount of time, even off treatment, and then all of a sudden they'll come into the emergency room in intense crisis, in kidney failure, right, with heavy rash and, potentially all kinds of things, right? And so, in MS, for example, which is another autoimmune disease, we don't necessarily remember that that's the case, right? Because in MS, we have this same relapsing remitting course, but the damage done during the relapse is permanent, right? So as we develop new plaques in the brain, it hopefully hits something that's not critical. But if it does hit something critical, you're very unlikely to regain that function. So even though your immune system has sort of stopped attacking for the time being, you're not necessarily going to gain back that function. So we sort of think...
Right, and I think the one way to think about this is we often talk about the sort of incorrect thinking of the immune system as just fights against invaders. Really, the way to think about it is trying to maintain homeostasis. And if the homeostasis is from an infection, then yeah, clearing the infection is part of getting back to homeostasis. But if the damage is from your immune system from inflammation, like that's pushing homeostasis or pushing away from homeostasis in a different direction. So then the immune system's role is to tamp down inflammation, right? And so as you're saying, in these autoimmune diseases, you have these situations where it's like the immune system thinks there's an insult, thinks there's an infectious thing that needs to get cleared. So the way to clear infections is to increase inflammation in various ways. Then we're pushing away from homeostasis in the other direction. In the case of something like TB, or in the case of an autoinflammatory disease, the infection, in quotes, never gets cleared. And so once you get far enough away from homeostasis in that direction, the immune system then pulls back because the inflammation is a problem, right? And so then we pull back, and then maybe we try to wall off the disease. Maybe in the case of autoimmunity, we're suppressing whatever T cells. But in that case, the insult never gets cleared because it can't, because it's self. And then so that's why you have these sort of you think of it like an oscillation where we keep trying to get back to some homeostatic line. We push in inflammation, then we pull that back and go in the other direction, then we come back. So we're sort of going in this sine curve way. And as you say, in some cases, as in MS, the damage is permanent. So in terms of function, we never come back to baseline. But in terms of the immune response, we're still in this cycle.
Yes, exactly. And so why am I mentioning all of that here, right? This is a mechanistic paper about RNA sensing in the innate system. And the reason I mention all of it is because of this oscillation, in most autoimmune diseases, you can't tell that somebody is sick the first time that system kicks off. Or if you do, you don't identify it necessarily as an autoimmune disorder, right? It frequently, and especially in lupus, it frequently takes multiple cycles over multiple years before you ever realize that the thing that's causing this patient to be sick is actually the progression of an inherent autoimmune disorder or chronic autoimmune disease, right? And so when we think about lupus, what we are thinking about is a disease state that is probably somewhere between 5 and 10 years in the making by the time the disease is actually diagnosed. So there is a profound disruption to the immune system that has been building over a decade in some patients. So when we're studying human lupus, right, what we are studying is not the initiation of an autoimmune disorder. It is the ongoing effector breakdown that characterizes chronic autoimmune disease, right? And so there's this huge gap in knowledge, right? And we know that that's true because there was a really clever study, right? So one of the most hallmark features of lupus is anti-double-stranded DNA, right? Like that's what everybody thinks about, right? This is one of the most stable features of the disease. What's interesting is double-stranded DNA, anti-double-stranded DNA, doesn't actually show up early in the autoimmune break. And that's known because of these interesting studies that were done out of the VA, basically, where what they did was they took patients who they had been, and these are military-aged people, right, who had received blood draws, who had gone through blood draws over the course of service, and all of those blood draws are banked, right? And then you go into that cohort and you pull out patients that, at some point develop lupus, diagnosed lupus, and then you go back into their historical record of serology and ask the question, when did the double-stranded DNA antibodies emerge? And the answer is usually years before the diagnosis of the disease. But what's more interesting than that is it's not just double-stranded DNA. Most of these patients, in fact, all these patients develop responses, autoantibody responses, against ribonucleate proteins and RNA years before the emergence of the DNA response, right? And it's much, much harder to measure, right? Anti-RNA ELISAs are a pain. That testing is much more tricky than anti-DNA responses. We typically don't see it show up in the literature, but it is well established now that the anti-RNA responses precede the anti-DNA responses, which is fascinating in and of itself, right? And the connection between the RNA, anti-RNA responses and the anti-ribonucleate protein responses, like anti-Smith, anti-Rho, those kinds of things, suggests that there is a deep connection between lupus, the emergence of lupus, and these responses against RNA molecules. Now, throwing in an additional piece, I told you that generally speaking, it is extraordinarily hard to study the beginning of disease, right? And that's largely true. But there has been some progress in that, especially over the last 3 or 4 years. And it came from genetic studies, and one in particular that I want to highlight, where there was a 7-year-old girl in Spain who developed rapid, aggressive onset lupus, right? And so what they did was they did some genomic studies, and what they found was there was a gain of function mutation in TLR7, which is the single-stranded RNA sensor, right? So what they did was
one of them,
one of the single-stranded RNA sensors. What they did was they basically took out that gain of function. They obviously tested, checked to see, what that mutation was doing. It turns out that there's a slight increase in binding affinity of that molecule to TLR7 or to RNA. And they took that and they put it into mice. And it turns out that that TLR7 gain of function is a monogenic driver of aggressive lupus, right? And since then, they've identified several other similar patients that have slight changes in TLR7 that have also been capable, seemingly, of driving this kind of lupus expression. Now, I would say that this does not hold probably for most patients, but it is fascinating that you can get a monogenic driver of the disease simply by, slightly increasing the affinity of TLR7 to RNA. And I would say beyond that, it has been shown now in a number of different labs, and we are using this model, that if you simply take a TLR7 agonist, we use R848, right, which is just a TLR7 molecule or agonist molecule, and you literally just paint it onto the skin of mice routinely, right, over the course of several weeks, they will develop autoantibody responses. They'll develop germinal centers. They'll do all the things that you'd expect a lupus-like mouse to do, which is fascinating, right? Because it means that the TLR7 RNA sensing threshold, whatever that is capable of skewing an entire immune response towards the autoreactive, right? It means that there is this careful
I think
level.
This is linked right to the phenomenon of epitope spreading, which is, if you think about a typical healthy person without autoimmunity, you do not have constant stimulation of immune receptors, right? Like, yeah, you've got a skin and a gut microbiome, but we've evolved to sort of like have ways of keeping immune responses suppressed for things that are separated from our internal tissues in various ways. We're getting sick all the time, especially as Matt and I are parents of young children, constant exposure to viruses. But it's not persistent in all of our tissues in quite the same way. And I think lupus in particular, unlike an autoimmune disease like MS, where you have it primarily localized to the central nervous system, even there you have epitope spreading, but lupus seems to be characterized by responses against things that are just everywhere. Like you mentioned, double-stranded DNA, everywhere. RNA, everywhere. Nuclear proteins, everywhere, right? Like every cell, well, not red blood cells maybe, but basically all tissues have all of these antigens. And what you often see in autoimmune diseases generally, but I think especially in lupus, because of the systemic nature of the antigens, the initial insult, whatever it is, the initial thing that triggered the immune response, it doesn't stay confined to that. Once you get breaks in tolerance against something that is self, it has a tendency to spread. And I think, we learned in graduate school that basically everybody has auto antibodies or autoimmune responses against something. Typically, we manage to keep it contained and we don't end up with that spreading. A hallmark of people that have disease that is measurable, that's clinically, clinically relevant, those people tend to have auto responses against lots of stuff. Probably it started with a single insult, but it seems to spread.
Yeah, or an environment, right, that promotes this sort of thing, right? And I think that we get caught up sometimes thinking that there must have been some single antigen that was the bad actor here, right? Like we somehow broke tolerance against a single antigen. I actually think that a more likely scenario in most cases is that you have developed an environment by which any autoimmune antigen is more likely to be responded against, right? And this is where we start to get into this idea of this TLR7 RNA tuning threshold, right? As we mess around with that threshold, it's not that a single antigen comes along and says, like, I'm a dominant autoimmune. It's that the system that normally selects against autoimmune responses in B cells starts to break down simply because the microenvironment is changing significantly, right? And we know that this is true in COVID. We know that under inflammatory conditions, we can get changes in the balance between well-tolerated and non-tolerated B cells. So another interesting phenomenon in autoimmune diseases, and particularly lupus, is that frequently, and I told you that, certainly the TLR7 is a monogenic driver of lupus, but there are plenty of other drivers that have been associated, or not drivers necessarily, but associates of disease. One is an increase in the amount of apoptotic bodies present in the blood, so there seems to be lack of clearance, and often that manifests as changes in the complement cascade, which doesn't allow for apoptotic bodies to be as efficiently taken up. There are increased nucleic acids frequently floating around in the blood of these patients, and all of this together, right, all of this literature together has sort of led people to believe that there is just a general increase in nucleic acid presence and inflammatory environmental factors coming together in the case of lupus that might be, sort of slowly driving this. And that's, I think, why this paper is particularly interesting, right? Because then what we're talking about is, how does the system, which, like you said, constantly hit with apoptotic bodies, right? Like all of us, all the time, right? Cells are apoptosing. There's DNA, there's RNA. Now, granted, we have DNases, we have RNases in our blood, and we talk about that a little bit in the paper. But, how does that system make sure that it routinely keeps that TLR7 threshold from sort of hitting that place where you're starting to get the initiation of those autoreactive responses? And I think what's interesting here is it turns out that there is an entirely different mechanism of how your immune system can prevent yourself from responding against these sort of circulating small RNAs. And hopefully maintain some level of homeostasis. And what's interesting is, like, none of what I just talked about really is in this paper.
I was going to say that it's interesting to listen to where your mind goes for an introduction here. And here you're going to see our bias. I feel like this paper really plays on each of our biases. You, lupus, adaptive immunity, all of that stuff. And when I read this paper, I'm really seeing, like, the history of innate immunity and, like, how that field grew, and thinking, like, more about the cell biology and location within cells as opposed to, like, the broader impacts. So I think that's a good place for me to start then, which is thinking about where this paper sits in the sort of innate immune landscape of research. And again, thinking back to this question of, like, why is this a Nature paper? I think implications for, the etiology, the beginnings of autoimmunity, I think is a really important thing. And that's certainly how they frame it. And I was thinking more in terms of where we stand right now in the innate immunity field. And we've talked about some of this history in the past, but just very broadly, early on, people knew about a lot of things that triggered the innate immune system. This was the insight of the original sort of Charlie Janeway hypothesis. We know that in order to get an adaptive immune response, you need to stimulate something else with adjuvants. Again, we've talked about the paper approaching the asymptote, where he sort of talks about this immunologist's dirty little secret. But we knew of all these molecules that stimulate inflammation. Lipopolysaccharide, nucleic acids, things like alum. So there's all of these different triggers of inflammation that were known. And very early on, the research was, what are the receptors for these things? How is the body sensing LPS? How is the body sensing nucleic acid? How is it sensing damage from these physical structures like alum, these aluminum salts that are damaging things? And so there was a long period where there were these sort of genetic explorations trying to find the receptors. First, we found cell surface receptors like TLRs. Some TLRs are also found in endosomes. So these are topologically facing the outside, but they're vesicles inside the cell. Then people started discovering intracellular receptors, things like RIG-I and MDA5 that detect RNA inside cells. Things like the inflammasome that are detecting other parts of cell state that can lead to apoptosis or pyroptosis, which is inflammatory cell death. Once all these receptors were found, a sort of another branch of like really high impact papers, once a bunch of receptors were known, was sort of learning more about the cell biology and the biochemistry of the signaling responses. So for example, downstream of TLRs, you have things like MIDI88, which is this critical adapter protein downstream of TLRs that is required for propagating that signal. Sort of learned about all of the things. Once the receptor binds, it then activates enzymes, it activates inflammatory signaling cascades that lead to different changes in gene expression. A bunch of different pathways were found here. We also talked about in a previous episode the sort of next phase to me, which was there weren't very many of these, but one example is cGAS and cGAMP, where a receptor and a brand new innate immune signaling molecule were identified. So this is like we found both a brand new ligand and a brand new receptor that triggered a lot of these same processes. So that was like a sort of new phase. We talked about, I think, pretty recent episode, we talked about sort of an extension of that where these small molecules are being able to passage between cells to trigger inflammation. So to me now, so we've got receptors, we've got cell biology and signaling, we've got receptor plus ligand. And to me, this paper is sort of like, it's a new inflammatory ligand plus some cell biology, but all the receptors are known. So that I was sort of thinking about that. Now we have this sort of slightly new phase where we're identifying brand new ligands, in particular ligands that are not pathogen associated, but self-associated. And now we have to protect from this ligand to prevent autoinflammation. Mm-hmm. And so that's kind of what this paper is about. So I wanted to mention also briefly, so the second to last author here, co-corresponding author, Ryan Flynn, who is I think he's at Harvard. So he's at Boston Children's. And his postdoc was at Stanford. He had a Cell paper in 2021 where he identified these new RNAs, essentially. So this is getting back to the cell biology piece. Again, this is some sort of basic cell and molecular biology, but folks probably know about, like, there's four major classes of biomolecules, right? There's proteins, lipids, carbohydrates, and nucleic acids. There's lots of subdivisions, obviously, within these. Within nucleic acid, there's RNA and DNA. And thinking about RNA, there's sort of the most commonly thought about RNA is messenger RNA. This is part of the central dogma of molecular biology, where you have a gene in DNA, gets transcribed into messenger RNA. This becomes the template to translate proteins, produce other molecules. Turns out there's lots and lots of different kinds of RNA. So transfer RNA is involved in translation. This is how amino acids get incorporated into developing protein chains. The ribosome, which is part of the machinery to make proteins, is what's called a riboprotein. It's this large structured molecule of RNA that complex with protein that's involved in translation. Turns out there's lots and lots of other kinds of RNAs. So microRNAs made a big splash, I think, just before we were in grad school, maybe late 90s, early 2000s. People started seeing these small RNAs that are regulatory. They can affect gene expression in various ways. A whole bunch of others that I know very little about, small nucleolar RNAs, SNO RNAs. In this paper, I learned just from the background yRNA, which I had never heard of before. So there's all these other kinds of RNAs, including what this guy Ryan Flynn discovered during, I assume, his postdoc, are these RNAs that are actually bound to the plasma membrane facing the outside of the cell. And what he also showed, in addition to just their location on the outside of the cell, these RNAs are glycosylated, meaning they have these sugar structures covalently attached to them. Glycosylation itself is not novel. We know that most cell surface proteins that are facing the outside of the cell are themselves glycosylated. So in the Golgi complex, as proteins are translated, whether they're membrane bound or secreted, as they move towards the outside of the cell, they get all of these sugar attachments. So the outside surface of a cell is essentially just a cloud of sugar that's complex to protein receptors, or apparently now, we previously also knew about glycolipids. So plasma membrane lipids also have sugars attached to them. And now we've got these RNAs, these structured RNAs that are plasma membrane bound, but have sugars attached to them. So that was the Cell paper in 2021, sorry. And now this paper is sort of more about the implications for those RNAs on the immune system. So that's kind of the primary background I wanted to talk through getting into this paper.
Since you had gone into that Cell paper, what was discussed as these things were being identified on the surface of the cell? So, we have all of these glycoproteins, and they're not just there. They all have some function, right? They're involved in metabolism, they're involved in cell signaling, they're involved in cell adhesion, they're involved in all of these things. And then you've got these nucleic acids, right? And we know that, RNA can be tricky in that, it can fold on itself frequently. Single-stranded RNA can sort of fold on itself in interesting shapes, and those shapes can kind of act like protein shapes. And so you can get many of the same functions that you can get in proteins, cell adhesion, recognition, things like this. Aptamer biology is an interesting way that field develops. But what did they say in that paper about what these RNAs might be for?
Yeah, I mean, I didn't read that paper in any great detail, if I'm being totally honest.
Okay.
But the primary implication, just like looking at the abstract and a little bit about the discussion, they were mostly talking about it in terms of, and they show this in a couple of the figures. They show that these sugars, they were sort of focused on the sugar piece of it. And so they showed, for example, that a bunch of cGLEX, these proteins that bind to sugars, often involved in cell adhesion, they sort of show the range of cGLEX that can bind to these sugars, and other lectins that might be implicated. So they were thinking about it, I think, and again, I didn't read in any great detail, but they were talking about it in terms of like, these are ligands for other cells to bind.
Yeah, it sounds like
for whatever reason. And they don't really, it's a very, very biochemistry and cell biology heavy paper. So it's really looking at the construction and sort of the passage through the Golgi, and they do all of this work to show like where you can degrade the sugars, which enzymes you can knock out to prevent sugar modifications, etc. So in this paper now that we're looking at, this Nature paper, they're getting into now the immune implications of this. And so it starts in figure 1, looking at the response to the immune system if you remove these sugars from RNA. So you've got cells, they're using HeLa cells, which is a very common tissue culture cell line in research. And basically they just treat it extracellularly with a enzyme that will degrade the sugar cap on these RNAs called PNGase. I wrote down somewhere, peptide N-glycosidase. And basically you remove those sugars. It's going to remove sugars from all the proteins as well, I think, on the cell surface. But basically if you treat HeLa cells with this enzyme, and then you put them in the presence of innate immune cells like macrophages, you get interferon beta produced. So interferon beta, classic response of both the toll-like receptors that sense nucleic acid, as well as intracellular receptors that detect nucleic acid. It's an antiviral response, right? So we see RNA that shouldn't be where it is. this is an indication we've got a virus present, and so you produce interferons.
Can I jump in real quick? And one of the most interesting exercises that we did in the paper reading course, I think, when we were grad students, and I'd encourage anybody out there that is sort of going through actually really any of the scientific PhDs out there. A really interesting way to think about a paper is distilling down to what specific panel, like if you were to pull the paper apart and you were to get rid of almost everything, what specific panel would have distilled the central concept? And it's really interesting in this paper because I think you could make an argument for a couple of them, but it is extraordinarily rare that figure 1A might be in the running for the most centralized panel, because we're going to spend now the rest of the paper talking about the different situations in which figure 1A remains true, and we'll even get into some mechanism. But I found it fascinating that basically like you get to figure 1B and the paper's basically over, right? Which is cool because
everything is icing on that particular cake. Yeah, exactly.
Right, and so like if you go back into the historical literature, and we bitch and moan about this all the time, right? As modern immunologists, right? You go back into the historic literature around immunology, and it's much more frequently the case in papers from the 60s, 70s, and 80s that this would be true, where you just like, bang, there's the observation, right? And now I'm going to spend a little bit of time talking about that observation and sort of where it is, but like it's right there. That's the first thing that we saw. So anyway, I thought that was cool. It's very, very rare that that's the case.
Yeah, absolutely.
Yeah.
So they show 1A is just if we treat with this PNGase, we get this interferon response. In these panels, the first panels, it's using raw macrophages, which is just an immortalized macrophage cell line. Later on, they basically do the same. It took me a while to figure out like what's the difference between these things, but later on, it's just a different kind of macrophage, peritoneal macrophages from mice. And you see that if you degrade sugars, you get an interferon beta response when in the presence of macrophages. If you treat also with RNAs, so you're degrading RNA, you lose that interferon response, which I think is kind of cool. They go on to show a little bit about the signaling pathway. So they show that IRF3 is activated, TBK1 is activated. This is, again, looking inside at the signaling cascade. IRF3 is downstream of the endosomal TLRs, like TLR7. They show more of the signaling cascade, in particular the MAP kinases. So we've got JNK activation, we've got ERK activation, again, in the presence of PNGase, not if you do also RNase as well. So this seems to be a function of those RNAs on the cell surface. And again, we're treating extracellularly. So we're treating the HeLa cells to remove sugars from the outside of those cells. And in principle, also degrading RNA, again, on the cell surface. These are external treatments. Again, in F, they show the same thing is true of peritoneal macrophages. In G, so then they want to say, like, okay, is this a cell intrinsic thing, or is this potentially also true externally? Like, where are these glycoRNAs coming from? So they show...
Sorry, real quick, before you jump in there, you mentioned HeLa cells. I don't think that we're at HeLa cells yet. I think that up at the top, we are looking at the introduction of soluble small RNAs that have been isolated.
You're totally right.
Yep.
Yeah.
And so we'll get to HeLa cells eventually. But
RNA, yeah, okay, so you're right.
Yep.
Yeah, so we've got different concentrations of small RNAs extracted from HeLa cells.
Correct.
So this is right. Primarily, the primary ligand here is the cell surface RNA, but it's extracted. It's not on the cell itself. You're totally right. So then they wanted to ask, are these RNAs like floating around? In G, they're showing it's present in both human and mouse sera. We see a bunch of these RNAs that have sugar modifications. And that serum in H through K, all of these things from both human and mouse If you treat with PNGase, you take off those sugars, you get an interferon response. If you also treat with RNase to remove the RNA itself or to break it down into nucleotides, you lose the response. So you seem to need these structured RNAs in the absence of the sugar modification in order to trigger macrophages to respond.
Right, which would beg the very obvious experiment, which I will not do in my lab, but somebody should do out in their labs that are also interested in autoimmune diseases. If you took an SLE patient, one, is there an elevated level of small RNAs as being shown here in the sera of normal, healthy human patients? And B, what is the ratio of glycosylated to non-glycosylated small RNAs in circulation, right?
Or more to the point, in those biobank samples, prior to SLE, fulminant SLE, do you have a difference in the ratio?
So there's your PhD project. You're welcome.
All right, awesome. So the next thing they do, so now they're moving into like actual full cells, and they want to look at, is there an implication for cells undergoing cell death? So when cells typically undergo apoptotic cell death, that is a non-inflammatory process. There is a version of cell death that is inflammatory called pyroptosis, but this is not that. Here we're inducing cells to commit suicide, and typically these cells are engulfed by macrophages. Macrophages love to eat apoptotic cells, but it's non-inflammatory process.
Right. Efferocytosis. Right.
Efferocytosis.
Yep. Goes right in the title.
Yeah. And what they do in figure 2 is they just, again, show that if we treat with PNGase, so we remove those sugars, we're getting a very strong interferon response. They do different ratios of apoptotic cells to macrophages in panel B. In panel C, they want to show that this is dependent on cell uptake. So they treat with a couple of drugs that inhibit endocytosis by the macrophages. And so they show that if you treat with cytokinin D or latrunculin B, then you prevent that interferon beta response. So this suggests that it's not detection at the cell surface. It's not any of the cell surface receptors. It's probably one of these endosomal receptors like TLR7. They haven't shown that directly yet. They also want to know, so in the first panels, they're using a drug treatment to induce apoptosis, but they want to see, are other forms of apoptosis, do they have the same effect? So they do UV, so where they basically shine UV light on the HeLa cells, cause lots of DNA damage that induces apoptosis. Again, same thing. We treat with PNGase, we get an interferon beta response. We also treat with RNase, we lose that response. So it's sort of variations on a theme here, sort of marching down the different explanations to see, do we still see the same effect? And the answer is yes. Figure 2E, again, we're just looking at intracellular signaling with IRF3 phosphorylation. In F, this was a little confusing again. In the text, they talk about raw macrophages again, but in the legend, it's peritoneal macrophages. So they're doing, again, the same thing. Previously, it was a macrophage cell line. Now they're doing primary cells from mice, from the peritoneum, and all the same stuff. Apoptotic cells, whether it's drug or UV, whatever ratio, treat with PNGase, you get interferon response. You treat with PNGase and RNase, you lose that response. So without the sugar cap, these RNAs are triggering interferon beta from apoptotic cells.
And one might ask why you need peritoneal macrophages on top of the raw macrophage lines and all of those things. And there's probably two answers to that. One is that you always want to confirm in primary cells that something, it's not just a weird artifact of, a raw macrophage line and all of that. But the more, I think, relevant technical reason that you would do this is because if you can do it in peritoneal macrophages, those are easy to isolate, and you can isolate them from a bunch of different mice of genetic backgrounds, which is ultimately where the papers got ahead in order to dig into mechanism, right? So you do it all the easy way in vitro to start, and then eventually you push that into the ex vivo systems that are actually going to allow you to manipulate the genetics.
Yeah, absolutely. All right. And then I don't have notes on G and H here. they're looking at IL-6 and TNF. So these are just other inflammatory cytokines.
THP1 macrophages, it looks like, after...
THP1, that's human macrophage cell line.
Yeah.
So
Yep.
raw max and peritoneal max are from mice. This is just, so THP1s are human macrophages.
Yep.
Human macrophage cell line. So showing this is not species specific. Cool. All right. So now, so we've shown in vitro that we have this response that seems to be triggered by these cell surface bound RNAs that have these sugar modifications. If we remove the sugar modification, we're getting interferon and a little bit of inflammation. So now they want to get into what specifically, or they want to talk more about, like, specifically what is it about these sugars and these RNAs that are inducing the response. And so they're now looking at these DTW D2 knockout animals. So these animals lack a particular RNA modification that is, I think it's the site of where the glycosylation happens, but it's a particular kind of modification called, where did I put it? It's got a weird structure. It's some particular nucleotide that has a chemical modification called ACP3U. So that's a uracil that has this particular chemical modification, chemical change that is different. And this was a feature of those glycoRNAs that were found in the previous paper. And this gene, DTWD2, is the gene that generates that modification. And so if you knock that out in animals, and then you try to do this same thing, you don't get the same interferon response. So they're suggesting there's something about this particular RNA modification that is the trigger that the receptor is binding to, or whatever, that somehow that modification is triggering the immune response. So they show there's two different genes that are involved here, or I guess they have two different knockout animals of this DTWD2. Mm-hmm. They show that in the absence of this modification, you lose the interferon response, even if you treat with PNGAs.
Yep.
Then, again, they're showing inflammatory cytokines. Here, in D&E, they're treating with a synthetic RNA ligand, or a synthetic RNA molecule that has this particular modification. And they show that at various concentrations, you are getting inflammatory responses. Here, they're looking at IL-6 instead of interferon for reasons that aren't immediately clear to me. But if the RNA is not modified, you don't get that response. If it is modified, they have two different versions of this compound, and you're getting the inflammatory response. They do RNA sequencing to show, or no, that's not RNA sequencing.
No, that's right. It is.
Is it?
This is actually, to be honest with you, this is the only part of this paper that I found disappointing.
Okay.
Because up until this
it's
point,
pathway analysis. Okay.
Yeah, it's a pathway analysis. Up until this point, I was thinking this is some old school biology, right? like this paper could have been written 20 years ago, right? And fine, like, I understand why you would do this experiment. I understand it's easy, and it's not single cell RNA sequencing, at least.
True.
So I'll give them that. It's just general RNA sequencing.
It's bulk RNA sequencing of the macrophages. But I'm sure a reviewer asked for this. It's just plopped in the middle.
Yeah, you had the mechanism already. You had the stimulatory molecule. You had probably downstream signaling already. Like, you had it, and they made you do it. And I don't like that it's there.
No, it's dumb. I agree. I'm sure a reviewer asked for this, because you're right. Like, if you're triggering TLR7, that pathway has been done to death. This is not different.
Right.
This is not different.
Right.
Yeah.
Anyway.
Totally agree. And then they're showing that If you have just free uracil, without the modification, you don't get interferon response, or this is TNF again. I don't know why they keep switching back to inflammatory cytokines. I mean, maybe it's just because those inflammatory cytokines are easier to measure. I don't know. I haven't done these assays in a very long time. If you modify, so again, these are sort of synthetic compounds, uracil by itself, no response. ACP3 uracil, yes response. If you bind a glucnac, which is one of these sugar modifications, sort of a very simple one, if you have the ACP3U with a glucnac attached to it, no response. So this is sort of distilling this down to the most basic component that you could imagine to show mechanistically it is this ACP3 uracil that is the trigger. If it's bound to sugar, you don't get the response. So the sugar is blocking whatever part of that molecule that the receptor binds to. And it's quite convincing responses here, in my opinion.
Yeah. No, I mean, there's really nothing controversial, literally, about anything in this paper. It's black and white.
It's just a really straightforward biochemical and cell biological march through the function here.
Yeah.
Then so in figure 4, now they're getting into, okay, what is the receptor specifically? Again, here they're looking at bone marrow-derived macrophages, which are primary cells, and peritoneal macrophages, and they're looking at different genetic backgrounds. So you knock out MyD88, which is critical for TLR responses. You lose some of that signal. You also knock out TRIF, which is downstream of TLR3, which is a different RNA sensing TLR. You get almost no response. So this is a bit interesting that so TRIF is not necessary for TLR7, but a TRIF knockout completely abolishes the response. MyD88 is not necessarily, it's necessary for TLR7, but not TLR3. You knock it out, you lose a lot of the response. So there's maybe some synergy going on here. They have a little bit of comment in the text that's not super satisfying, in my opinion. Similarly, in panel C, if you knock out either TLR3 or TLR7, you lose almost all of the interferon response, but not all of it. So again, maybe some synergy going on here that's not totally clear. So that's panels C and D. What's the difference between C and D? I don't remember. It's probably just the two different cell types again. We're probably looking at BMDMs versus peritoneal max. There. BMs.
Yep, peritoneal max. Yes. D is peritoneal max, C is BMDMs.
Okay.
Yep.
Great. So again, two different cell types. And then panel E. So panels A through D here, we're looking at these small RNAs extracts. E, they look again at apoptotic cells. Same thing. You lose TLR3, you lose TLR7, you get drastic reductions. And then in F, they're doing transfection essentially. So they're using lipofectamine to put in these RNAs, and they show that you need the modified small RNAs, and again, you lose the response with TLR3 and TLR7. So it's not, it can't be the even if you transfect, it's not the intracellular, like RIG-I MDA5 kinds of responses. You still lose the response entirely if you don't have TLR3 and TLR7. So this, I think, very strong evidence with MyD88 MyD88 and TRIF knockouts, TLR3 and TLR7 knockouts. You're losing a huge amount of this response, and so clearly it's the cell surface receptors that matter here. And that's the paper. I mean, there's lots of supplementary figures too, but and then they have a nice little schematic.
Yeah, it's nice actually. They did a good job with that.
Yeah, I like it. And that's the paper. I mean, it's very solid. Again, the biology is solid. I came away from this thinking this is primarily a Nature paper because, again, this is a brand new ligand, self-ligand, and regulatory mechanism that hadn't previously been described. The implications for autoimmunity, as you said at the beginning, fairly obvious. I mean, not obvious in the sense that, like, a lot of work still needs to be done to nail this down, and probably autoimmune responses are pretty complex, and so this is unlikely to explain all lupus or other autoimmunity, but it is a very clear, very convincing trigger and some cool cell biology. I didn't know we had RNA sticking to our cell surface. Like, that's weird.
Sure. Yeah.
And that's cool. So the previous paper showed that, but this is showing like very clear implications for the immune system. So yeah, cool paper.
No, I like it.
I liked it a lot.
I think it's nice, simple, straightforward, to the point.
You would hate the cell paper, the 2021 paper. It's all gels. It's like, it's Southern blots, it's Western blots, it's tons and tons. It's just gel after gel after gel, showing all this stuff. Very, very well done, but you would hate it.
My graduate student came up to me the other day, and he's like, we have to do this transfection. We're working on a lipid nanoparticle-based mRNA vaccine. We're trying to wrap up a paper pretty quick. And he's like, we have to test to make sure that we're getting expression levels. And I was like, yeah, I mean, we could run an ELISA. He's like, we could do a Western blot. And I was like, I can't support that.
Send him to Abby for that.
What's that?
You need to do the Western blot, Matt. Come on.
No, I know.
You can't just look at RNA expression.
I know. But, yep. Anyway, I hate Western blots.
Yes.
Yeah.
We've been through that story many times, so I won't tell it again.
No.
So, yeah, I mean, this paper, I feel like I have nothing bad to say about this paper. It's incredibly thorough, very cool biology, just solid.
Yeah.
Solid paper.
Yeah, and it'll be interesting, I think, because it feels like the beginning of a new sort of way of looking at RNA, right? And I think that's probably a piece of this. I mean, clearly the Cell paper was the first version of that. But I think, taking this novel finding of RNA stuck to the surface of these cells for like reasons that are completely unclear, right? Because I think, have always assumed that all of the functional bits of the cell out on the surface are proteins, right?
Yeah.
And so I think
and there's the explanation that this is like a binding platform for cGAS. Like, that's not satisfying, because you could just have the same sugar modifications on proteins too, or lipids even. I mean, there's no reason that you would need RNA to serve this function if it's just for the sugar. So there must be some other function of these RNAs. Yeah, the sugar is protecting from autoimmunity here, or autoinflammation, but I don't think the sugar is the point.
Yeah,
because basically everything at the cell surface is glycosylated.
Right, exactly.
So what is the RNA doing out there, dangling off the plasma membrane?
I don't know, but I'm certain that the Flynn group will continue to tell us, and we'll be interested to learn, I think, as we go. And cool paper. Yeah, we liked it.
This has been Audiommunity, episode 47. I have been reaching out to folks. Matt and I have been talking about this for ages, that we really want to start bringing some guests on. So I have a couple of people potentially in the queue, including one that's going to be a stretch for the immune system, but a very, very cool paper that I think we should look at anyway. So stay tuned for that. In the future, if you like this episode and the stuff that we do, please support us. There's lots of ways you can do it. We've talked about Patreon in the past, patreon.com/Audiommunity. You can like and subscribe the podcast on whatever podcatching service you use. Our website, audiommunity.org. You can find all of our past episodes. You can find videos on YouTube, youtube.com/@Audiommunity. But as we keep saying, the best way to support the show is to share us with your friends. tell another grad student or another postdoc about the show. If you have questions and comments, you can email us at contact@audiomunity.org. And the music at the beginning and the end of the show is composed by Rachel Reinick. And we'll see you next time.
Thanks, y'all.
Sometimes you get lucky.